All right, thank you very much. The chatter ahead, I think I'm trying to convince myself, I hope all of you are working on this too, that remember, this is fun, right? You get to go meet people you don't see every day, and you do presentations in person, and yes, sometimes it's a hassle to travel and that kind of thing, but this is fun. And it's good to see people, and I certainly appreciate the invitation. When I was thinking about what to talk about, some of what I'm going to talk about today is based on the talk I gave last fall, but I am not I don't have any expertise in design of policy. I was president of the American Society of Agronomy for 3 long years, including most of the COVID years, as well as the years where we lost, fired, and then re-lost our CEO. And so I spent a lot of time struggling with things related to representing our science to those that we think should be listening to us, but don't necessarily seem to be hearing our message. And so some of what I'm going to say is reflections on that, because the whole situation we find ourselves in with climate smart commodities, maybe you don't feel this way, but I feel, sometimes I look around and I'm like, how did we get here? Some of the things we know better. We know this. We know this may or may not be a good idea. We know what to do, but in some cases, nobody seems to be hearing us. And so it's not just for policy, it's perhaps our more traditional stakeholders as well. And now I'm trying to advance the slides. I'd like to say, go ahead, slides. There we go.
The feedback is that they're hearing you best when you're in front of your computer. Okay. So
then it must be, okay, so it must be going, because it's going through the computer as well. Okay, I will try to stay behind the desk. It's a little high for me. All right. So when I was president of, let me see how far ahead. Okay, I'm good. When I was president of ASA towards the end of my year as president and into my post-presidential year, I got a phone call from my old major professor. If you're lucky, they never go away. And he said he'd been reading the Wall Street Journal, and he's like, there's some crazy ideas that are out there about what we should be doing in agriculture relative to climate change. And we need to get a different message out there. The societies need to get a better message out there. And so this led to the decision to survey our membership and simply ask them questions around, it was really motivated by the soil health management as a natural solution to climate. And what did our members think when it came to mitigation and adaptation? And we knew we were going to get some sort of response that it's complicated. Some of you may have I hope, took the survey. As with many things in our societies, we ask you to do things and we get about 8 to 14% response from our membership. So maybe a few people did things and respond to this, but we asked them to rank big bucket strategies. And there was a small group with our science policy office. Our professional societies have a small science policy office that is tasked with communicating our message to all policymakers, which is an enormous task. And the results came out clearly like this, pretty clearly, that there were 4 strategies that our membership, and this is crops, soils, and agronomy, thought were important. There were a few that they clearly thought were unimportant, and then some in between, and protecting or enhancing the soil was at the top. But when we asked them to parse out this issue of adaptation versus mitigation, we didn't get anywhere near as clear an answer. We knew we wouldn't. We didn't necessarily design the survey to do that. most of them indicated that they were responding in the context of adaptation or both adaptation and mitigation. So if they were thinking about adaptation or adaptation and mitigation, protecting and enhancing the soil was at the top, improved nutrient management was number 4. But if they were thinking about this issue of mitigation or both, improved nutrient management moved up. Other, are you dealing with admitting people who are, okay. A few other tidbits was that most people in our professional societies felt they were responding in the context of adaptation or some generalized picture, and really didn't focus on mitigation, even though that was the topic that was sort of swirling around in the ethos of the discussions of policies. Let me move this so you all can see. I tend to fill my slide. And in the end, most of the time frames were pretty long, and soil health was the longest. Okay. Advance. That's weird. Okay. So this issue, it didn't come from nowhere, and it's easy to see now, but to a certain extent, it did seem to almost come from, where did this come from? That all of a sudden, I have in my state, people calling, legislators calling me and asking me about whether agriculture is a natural solution to climate change. Now, remember, we're a state that wants to burn coal. So that's the framing of the can we do this? Is this a solution? Can we do this and then keep burning coal? So where did this come from? Well, there has been a very concerted effort on the part of many people probably involved with the initial year of soils with FAO, trying to get the year of soils. Yeah, okay. And then after that came soil health, trying to raise awareness of soil health. And we, being the human beings that we are, we love the concepts that anthropomorphize things we otherwise don't think about. So now soil's healthy also. And it's a great branding campaign. Everybody's talking about soil health. That's great. And then Hollywood's talking about soil health, and it's all wonderful, and you can buy wine. And then we have this thing called the climate smart commodities, and suddenly it is our policy, and we're pushing billions of dollars into climate mitigation to the tune of, I think it's now 3.3 or more billion dollars have gone into the climate smart commodities program. Okay, so a lot of is the program correctly directed? Are they targeting the right thing? Lots of hand wringing, and then lots of discussion of why wasn't agronomy at the table to help design the program? Where were crops and soil sciences? And so I view agronomy, medicine is not the only discipline where you have clinicians. Agronomists work with the real world. Crop and soil, the science of crop and soil management, you know how it's going to be realized, you know it's not on the lab bench, and we have always been part of understanding how something is going to work in the real world, and therefore realistically we should be out there not just advising farmers, but we should be advising policymakers. And for some reason that wasn't seeming to stick. This again goes back to our survey about whether we needed more knowledge. Most people thought that it was things like economics, more knowledge or more research, the sort of, not the hottest colors, not cool, but everybody isn't saying we need more research on this. We know a lot of stuff. we scientists think we know a lot of stuff. Maybe for crop improvement, we need more. Sort of nutrient management is across the board. CCAs thought it was either knowledge or economics. They didn't make the link between the economics and policy, but that's okay. Nonetheless, we're in a situation where if you look at where we've been with the science and what our messaging has tended to be in the context of extension, it's been more about the science of adaptation and what we know. Yeah, increase organic matter. Here are the benefits you'll get from increasing organic matter. Here's why it will help you on your farm and why you should adopt strategies. And then when you look at the policy orientation, it's do it so you can get a carbon credit that you'll get paid for. And we can create the markets to do it. So fairly different spots. Science is saying it's a natural solution to adaptation. Policy, it's a natural solution to mitigation. And I'm not sure there's much conversation in between. So in all of that, lot of reflection and discussion, and on my part, thinking about why aren't we there? Have we hidden our light under a bushel? Are we not talking to the right people? And to a certain extent, that's true. If you look at what I have told my students to do, what I have done myself, I have an extension role. My role is to deliver recommendations to farmers. I work on the science to support that. I do that. I might publish in Soil Science Society of America journal, fine journal, until recently, primarily not open access, so read primarily by members. We might get something featured in CSA News as a cool study, subscription only, no policymaker will read it. And then we might get it put in Crops and Soils, which is directed to our certified crop advisors. We don't think about the role we might play going and explaining science to policymakers. I will say that this is not necessarily comfortable, and you can't just, you don't necessarily just call somebody up and say, oh, do you want to have a policymaker meeting tonight at, at sundown? There's a whole lot of layers you go through, and that's why we have our science policy office, but we have not historically done a lot of this, they admit. And Michael, you too can get to do this. It's really fun, and you better get a good night's sleep before you do this kind of thing. But we haven't done that, so we haven't been sending the message. We don't necessarily publish in some of these higher end journals. We had a discussion about this in the past. I'll tell you, if I submit my stuff to Science, they're not going to publish it for a variety of reasons. One of which is that if you look at who's on the editorial board of Science, they won't have any understanding of what I'm doing and why. So, okay, it's not targeting it. But the thing to know about Science and to think about is they have a promotion machine where they don't wait for you to promote. They pick pieces and they promote it, and it gets into the New York Times or the Wall Street Journal. And we don't have those kinds of mechanisms for the outlets that we publish in. Okay, so who else doesn't know that agronomists underpin the climate science research? We're the clinicians. We've been out there. We are the scientists for on-the-ground climate change, adaptation, mitigation in not 10 years from now, right now. So who knows we're there? Well, Science doesn't know we're there. So this is a Scopus analysis of climate change, and all most important, top 10 most important or cited climate change papers. They're either about the climate itself at various scales, or they're about natural systems. They're not about agriculture. And then if you look at how we have branded our science, how we have aligned it, I don't think it occurred to many of us to say, oh, I'm doing science that's important for climate change adaptation. I'm doing science to help with nutrient management. I'm not going to say, brand it, tag it in a way that it is considered to be an identifiable when somebody goes to look for it as a climate change reference. And so when you search something like Scopus for papers on agriculture in the climate change area. I'm not saying that the staffers for for policymakers do this. Some do, but we're not there. And this is just looks back, 1990 through 2014. We really weren't very much present. If you look in the past year, climate change and agriculture, most of the references, about a 16% reference of agriculture in those articles. If you actually try to drill it down to agronomy itself, it's only 459. We can have the debate on whether we should rename agronomy department something else, because nobody knows what agronomy is. And then you add carbon and you get even fewer references, and you add mitigate or sequestration, and it's not tagged at all. Okay, it's not tagged. Does that matter? Well, it's debatable, maybe, maybe not. But other scientists who have the ability who are at the table in these discussions, if they don't see our science where they look, they're not going to say, oh, we need an agronomist at the table when we're talking about these big policy issues. So it's a problem. Science doesn't know we're there. Another of my points, and I wish I could get this to go away completely, but I'm not sure I can. We are looking now at these targets, these measurable impacts that are small relative to variability, slowly accruing, and when you try to measure them, the effects are highly variable. And so this is a challenge to measuring and monitoring in the real world. I think you can get your head around that. It's a real challenge to our culture of how we do science. Why does it suddenly decide it doesn't want to advance? Seems to be every time I move that bar.
Well, you can hide.
Oh, it's for some reason I'm just trying to advance the. I'll hide this, but I was just trying to. Yeah, and oops.
For the floating ones.
Where is it?
There you go.
Yeah. Okay. So if you think about some of the language that you may have heard about what would be beneficial, and oftentimes you hear this 10%, if we could just increase soil carbon by 10%, that would be great. It would solve 30 years of anthropogenic soil CO2 emissions, and we would, could credit that against other carbon emissions. And the reflection, we should know, based on our existing science, how hard this would be to do. And we should know and be able to articulate reasonable expected magnitudes of change, what we can expect to observe at scale, that kind of thing, which practices may or may not have measurable effects. So the challenge for us is that when we do our research the way we have always done it, we move beyond the branding and thinking about other issues with our science. We're going after these small effect sizes, and we tend to have to do or want to do studies that are generally underpowered for identifying these effects. Even if you have a long time horizon and you've got 2 plots that have been in place for 20 years, you may not have enough statistical power in your average experimental design. Just thinking about it, historically, what we have done is this comes out of, the Rothamsted, the development of Fisherian statistics, how we went about doing agricultural field research, lots of small plots, but 4 or 5 replicates with Fisherian statistics. We got into replication, randomization, local control to eliminate the heterogeneity we didn't want to look at. We didn't always want to look at the covariance. We were more targeted on that effect sizes. So we've got small teams working on this. We've got a hypothesis. And that type of science has made huge advances, but they have been where effect sizes are really large. So some of the initial advantage, does fertilizer work? Well, by golly, it does. And so, you know, if it's no fertilizer, but or quite a bit of fertilizer, you're going to get a big effect, and it's going to be pretty consistent, especially if you're in a lower fertility environment where many started. If you develop an improved line, and you've got a management package that goes with it, and suddenly you no longer have strata, you have a big difference. You have a big effect to make. Statistical designs themselves are a separate issue. I'm not going to get into it, but I think it's a really interesting discussion topic as we think about what we might do with on-farm collaborators, farm data, etc. My related point, and I've already mentioned this, is that the time to observe a change is often really long, and it's not relevant to farmers, many farmers, because they got to think about tomorrow, especially if they're renting. So today's farmers, some of the changes are not that relevant. So if you start talking to them about doing something today to get a change 15 years from now, and you're going to implement a policy to do that, well, I don't even know how you do it. But it's just, it boggles the mind in some ways to think about the disconnect between the objective, which may be fine, but the context in which that objective is going to have to be realized. And so just as a way of an example, I'm going to use some of my own data, but there's so much data out there that it illustrates this exact same point. So we have an experiment where we compare a reestablished native prairie to corn in a variety of systems. And I had a grad student who back in 2006, 12 years afterwards, did the whole total profile C stuff, and only saw little differences in concentration in the surface when compared to continuous corn or corn soy. And the total profile, no difference, but lots of variability. And in this case, this was samples collected in 1 year, 4 replicates, whatever. And so USDA, they're wanting to pay to incentivize for performance and not for practice. And Mike Castellano and I had a discussion about how did we get to that point, because we probably in the end are going to go back to paying for practice, not performance, because we're going to pay for practice with an assumed performance or a model performance that may be based on a fairly poorly calibrated model. But in the end, you know, this business has led us down the pathway of we have to be able to measure and assess this change at scale in the real world. And so if you think about that previous study I showed you, and that you take that sort of variability, and instead of thinking about, you know, a total soil profile, you think perhaps about a 30 centimeter layer, and you want to see a 10% change, but you had the same degree of variability. So you want to increase, in this case, 10% in a concentration as an example. You can do power analysis, right? Everybody knows how to do a power analysis? Power analysis, know how big your study size ought to be? Well, it's got to be a whole lot bigger than 4 replicates with that kind of variability and measure. So this is just shows you the number of samples you would need to find for carbon, and then also for bulk density. Now, I didn't do a literature search on the bulk density CVs, but they can be pretty darn high. I was just looking at some of my own measurements. And so that tells you I don't have a strong enough power. So, you know, I didn't find significance, but that doesn't necessarily mean there was an absence of effect. I just didn't have a powerful enough study. We also know, and have known for years, what to expect in a commercial testing lab. This is the North American Proficiency Testing Program. If they're going to be running, if commercial labs are going to be running samples, what is it reasonable to expect for them in terms of precision and accuracy? And for carbon, which they don't yet have combustion as a practice that they approve on or certify on, but they're talking to the Soil Health Institute about adding that, but 10%. The very change you want to see. Okay, so whatever. And all right, I'm doing I'm doing a lab experiment. So now I go back to thinking about my numbers, and I'm a scientist, and we know the sources of variation. We've been talking about them for years when we were talking about a sample you bring to a laboratory. And so presumably in my research, I've minimized this issue of selecting where I sample. I'm processing the sample correctly. I'm doing internal controls to know that I've run the analysis in the lab correctly. So then it comes down to, how did I interpret that lack of significance? Nothing happened, or I couldn't see it. And actually back then I thought about it as nothing maybe happened or hadn't happened yet, because we were only 10 years into it. I didn't know. But that got me thinking. And so I have concentration measurements now by combustion going back to 2012. And I just asked myself the question, okay, how many of these years do I need to put together to get a significant, to see if there's a difference between two of the treatments? And you can see from this is looking at the effect, the difference between continuous corn and restored prairie. Managed continuous corn, managed with regular N applications. No single year is there a difference, significant difference amongst those four. But across all of those 9 years that I have, it's just getting there to a significant, I have enough samples. And if you look at how, this is just a little illustration of how I get to that p-value. It took over all of those 9 years, which is exactly in line with power analysis. But let's go back and think about this a little bit more, because most people, you know, you're going to do a stock measurement. You're not going to do stock measurements over and over again. And most people aren't going to have a 27 rep study. So you want to put studies together. So how do we put studies together for a better understanding of small effect size? And again, this is where I get a little bit annoyed. So, you know, I love this quote. You know, how do we use our statistics? I didn't think about it back then so much, but those statistics told me, you know, you can do a power analysis on studies for meta-analysis just the way you can do it on studies for sample sizes. It's just a variation. And so am I making a type 2 error in saying there's no difference? There is a difference, I just don't have the power. And so the you know, add-on case to that, and I put it here, somebody can go back and look at it if they want. You have to make a bunch of assumptions, and I made all the simplest ones possible. All studies are like my study. And in the best case scenario, I wanted to look for that 10% difference. How many studies would I need to get to an 80 or 90% probability that I'm not, that I'm correctly rejecting a false null hypothesis? In other words, there's effect there. And I need probably 8 to 10 2-year rep studies of 4 replicates each year. And that's often what you see in the meta-analysis. You need to have that many studies. This is the type of variability we're looking at. Okay, what are some other issues when we get to trying to figure out what the heck is going on? Okay. There is positive bias in the literature. So what is positive bias? You know this. If you've been in science long enough, you are way more excited about the study where you got a significant result than the one that you got a non-significant result in. And what are you pushing to publish? You're pushing to publish the one that you got a significant result in. And the other one, well, you don't know what to do with it. And it's probably, you know, you're not that interested. It's novel, it's messy. Our own journals won't necessarily publish it because it's not novel and it's not, you know, that interesting. So no place there. I can't get it in there. This is us. I already said this. We just love that, you know, we not only want significance, we have an allegiance to that 0.05. And when we're talking about high variability things, we should probably rethink that P.05 thing. What are we really saying about that? And then if you're actually in extension, and again, I was talking a little bit to Mike about this because it's sort of been one of my tensions in extension. When I joined, there was no, basically I was told it was a waste of time to worry about publishing in peer-reviewed journals. I needed to get stuff out into extension bulletins and be out there talking. I would have to publish a few papers to get promoted. Problem is, you don't publish it, it's lost. And what's happening now is people are doing exactly what I could do to see whether or not it's a power issue with my small study size. They're putting studies together. So meta-analyses are taking off. Everybody wants to do a meta-analysis. So this is just another little search on meta-analysis in agronomy. And you can see that it's skyrocketing. Everybody is doing these. And if your data aren't in a peer-reviewed publication, I'm pretty darn sure nobody's bothering to try to get them into their meta-analysis. And so, you know, your results are here, and I have actually my predecessor's results at Purdue, and I even have the Bargle plots, the raw data from the Bargle Mingle or Stan Barber in my office. And I'm waiting for somebody to decide because I can't bear to pitch the things. But so the result is that when you're looking at the effect of something we would now call climate smart agriculture, you are looking at the positive effects or showing an effect is being overrepresented in the literature. So a systematic review will say something is likely to work better than it is in reality because of everything that gets left behind. I will add that the overall quality, and Sarah McMillan, who stepped into the background, has been sitting in on some of our discussions in a class. The overall quality of the SR systematic reviews and meta-analyses that are being done in agriculture is not that hot right now. And this is a methodology. It is a research methodology. We're doing our best to try to teach it. But the common problems, so what I'm showing here is that, you know, okay, people are calling them all kinds of things. Some use meta-analyses. Some say they're doing a systematic review. Somewhere in between, people think they're doing both. There's a standard methodology for doing it. It comes out of medicine and education. People really aren't following it. There's some key things you need to do so that your searching is repeatable. There's also a persistence of vote counting in the published literature, including, I mean, in our own journals. This year, I saw a major summary of the impacts of cover crops. I think it was on Soil C that counted non-significant article as a zero effect and used the zero effect in multiple regression, in regression analysis. It is not a zero effect. A zero is like, there's no difference. There's truly no difference. And so that's tremendously problematic to represent something as a zero value. It would be better just to leave it out and say these were non-significant studies, but they showed an effect. It just wasn't a P of 0.05. And there's lots of trouble with data extraction and trying to figure out how to teach this. I only have one or two more points about science and then just a couple more on the policy issues. Okay, proxy measures. If you work in, you know, working with farmers, the soil testing, most of our soil tests are indexed. They're things we figured out represent or correlate to the availability of something to something else. So that the test is a diagnostic. It diagnoses a condition, right? And we've always used things that we come down that are compromises. They don't perfectly represent plant available K, but they do a good enough job and they're cheap and efficient and whatever, and they're, you know, they're enough, precise enough and accurate enough. So this got me thinking about organic matter versus SOC measurements. My hypothesis in looking at numbers, at my own numbers, is neither is precise. There may be a trade-off in accuracy. SOC may be more accurate depending on what your objective is, but neither of them are not that precise, and I'll show you a little bit of data. And we need large numbers. And if we're thinking about what do we already know and how are we going to move ahead, we have, I don't know how many numbers on organic matter. This is just one farmer's field, a farmer collaborator. So organic matter, almost 9,982 organic matter measurements on 120 acre field. Okay, that's a lot of information and also a lot of variability, as you can see, in that range. So maybe you just pick one spot and pick another spot and get paid a whole lot of money. So with the protocol we're going to use in the field, we have to remember not to let the perfect be the enemy of pretty good. And we need to think about what that pretty good is. There's science, and then there's the diagnostic. And the diagnostic doesn't have to, it can be a proxy for what we think we need to do in science. As long as it's a good one, then we're okay. And so, you know, what should a soil test be? It should be rapid and accurate. The soil health big paper that they put out, they agree. If you look at the prevailing opinion, it's that LOI, loss on admission, the way we do it for commercial testing, isn't good enough. And so, you know, if the soil health handbook does talk about both LOI and dry combustion, because LOI is so much cheaper and done so much more frequently. I would debate this accuracy issue. This comes from their supplement, the Soil Health Institute's paper, supplementary material, where they actually concluded, although conversion factors are different depending on the temperature, so that's an issue. And you've got this issue of clay. That being said, you know, if you don't have, if it's not SOC analysis is not available, go with LOI, better than nothing. Okay, they agree with that. The other tidbit that's in there, one sentence, both LOI measurements and responded the same way as this SOC measurement to the effects they were looking at, except for rotation, where they noticed a slight difference where organic matter was significantly negative for LOI, but it wasn't for SOC. But look how close those values are. I don't know what you want. Those numbers numerically are really close in terms. So I went back, I compared my historic organic matter numbers, converted them to carbon using the quick and dirty factor everybody hates, and just said, okay, here I'm looking at my effect. And the red is my soil organic matter number in the top 20 centimeters. And my black dot is the carbon measurements we started making when we got a CN analyzer. Couldn't do them before. And if I look at the size of the effects, in most cases, we don't see an effect in organic matter in the first 2 years, first 2 decades. Those were when we were just doing organic matter. Then we do both, and both find an effect of similar magnitude. And I have 1 year that's really weird. Don't know why. I'm missing a year. I can add it back. Bottom line is they're showing me essentially what the Soil Health Institute said it would show. They're showing me the same effect. Now, I can't calculate the stock, but I'm seeing the effect. So you can ask yourself, and this just goes through the same exercise, what do you see if you add one? It looks exactly the same. We're seeing the same effect. The effect sizes are similar, the timelines, the numbers of samples, blah, blah, blah. So when you're coming to the lab protocols for commercial use or use at scale, are we asking or have we asked the right question? And I'm not sure that we have. We can look at this, you know, adaptation versus mitigation and what are we trying to do. If you ask the question, if you're asking the question about stock and change from the time I'm measuring it. So today I'm going to say you change it, I'll give you money. Okay, SOC. But if I'm simply trying to say, am I doing something that does a better job? Maybe organic matter is good enough and it's cheaper. So, okay. I did a quick search trying to see if anybody had compared this 1-to-1 for looking at that effect size. And I only came up with 2 studies that actually compared the 2 measurements in a way that you could see if they measured the same effect size. So that's an opportunity. There were lots of irrelevant studies and things like that, but I only found 2. And it wasn't enough to confirm or whatever. So we could go back and say, what can we do? Well, we could go look at lots of people out there in the literature suggest, why don't you just go now and look at some archive samples and measure SOC and see whether it changes. We could go back to the Soil Health Institute and say, okay, can we have another conversation about LOI in terms of responding, measuring response to the, adding a soil health practice. We need to establish targets if we're going to use something in any kind of diagnostic way. So a conversation I had with John earlier today. My analogy was, it's like, if we don't, we can say to people, okay, if you get a fever and you're sick, here's a thermometer and you can measure whether you're sick and then you can measure whether you're healthy. But if you don't know what your temperature ought to be, what's the point of having a thermometer, right? And that's kind of where we are right now, right? Go do these measurements and then I don't know what, right? So this science is telling science something, but it isn't necessarily telling us anything about a diagnostic. And so we need to develop diagnostics. We need to do some things differently with them. There's a whole raft of approaches to understanding, testing accuracy, sensitivity, specificity, predictive values, likelihood ratios in medicine. We can use them. Publication bias is out there. It's a problem. If you look in the gray literature, oftentimes stuff is messy. So this is, us keeping stuff in our file drawers. Last point on the science is get everything out there in the open. It's coming anyways. But this is a little analysis of our own journals that Jeff, he's online, did. But if you publish open access, you get cited more faster. So just publish open access. It's good for you. It's good for science, whatever. And publish your data. And that's a separate conversation, but in soil testing, we've always known we need a lot of numbers. It was never meant to be a small article, a small science exercise. Lots of scientists were supposed to do it together and combine their data sets. That goes way back to the 40s. We just have better tools to do it. So you can get away, people can understand what you're doing with your p-values. You can get your data to modelers if you publish your data. Again, for this, the modeling of soil carbon, I think the question, how much data, whatever. Last thoughts on the policy side of things. Okay, when we have scabs of money out there, do we use it well? And do we challenge ourselves? Or do we double down on things we've already said because we know it sells and we're too afraid to say, no, actually, I don't think this is working quite the way it ought to do it. And I'm picking on genetics right now, but Ken Passman in the group at Nebraska wrote an article that said genetics isn't everything. That if you look at why our yields have gotten higher in recent years, it's actually just because our climate has become more favorable. And that got into the National Geographic, and I got an email within a day of publication of the National Geographic from a provost to our dean to me, from a concerned supporter of the wondrous genetics research at Purdue that said, this can't be. Genetics is responsible for so much or whatever. I'm just picking on this example, and we had to go back, but basically, it called those who said genetics. There was the accusation out there that if you're saying it isn't genetics, you're attacking science. We don't seem to be able to have a rational conversation about what we know. And when you're a hammer, everything's a nail. And this extends to when you're talking to farmers. I've seen it, and you tend to do it yourself. This is what I know. This is what I think. This is what you should do. It goes to policymakers, especially when you're asking them for money, right? this is the most important thing. It is extremely difficult to get a nuanced message from a large group of scientists to send to policymakers. The best we can seem to come up with is a generalized statement on the global research needs for all of our scientists, which doesn't help them, and then say, you just got to give us more. And that's not very helpful to them. I don't have an answer, but I think, there are windows, and I'll go back to, I had the opportunity to testify. I was asked afterwards very specific questions that they needed feedback on. And one of them was doubling down on existing farm programs. Is that the way to go, or do we need something else? And I gave them some feedback, and I hit my favorite one about being able to synthesize whatever. But you need to be participating in conversations where we are providing rational guidance on the pathway forward and threading the needle. We've got, it's great. We don't want to dampen their enthusiasm for science and for agriculture and for soils and all that kind of thing. But we also don't want to be, I love this article. So, if you have a moment, go read this Amundsen perspectives piece on Kiss the Ground and Make a Wish, Soil Science in Hollywood. It's great. It ends by pointing out we have to be our own guardians of the messages that we are sending, or we will get bitten when it doesn't deliver on this global greatness that we sold them on. And that's a little bit my concern about what's happening. I heard a lot about moonshot language, and I find it I'm a little weary. And I actually Googled, who's talking moonshot language? Well, cancer's talking it again and whatever. And there's some perspectives out there on whether it's helpful to cancer. And some of them are sort of, if the money's well spent, then it's maybe a useful metaphor. To criticisms, one treatment fits all approach. It's continuing to keep people from understanding the reality of the challenge. And then, some, problems with what it conveys. And then, is it even relevant? And I guess I come down, when we talk, we need a soils cancer moonshot, or we need a soils moonshot. What does that mean? We have to give more useful advice than that. You need to be prepared to explain local losers and winners, and so what yields might go down, what yields might go up, why, and in lay terms. Yes, soybean yields have gone up. Is it because they're C3s and CO2, or is it because we've got better genetics? My last comment is about precision agriculture, which brings it back to precision and accuracy. I came to Purdue at the dawn of precision agriculture. We use the term all the time. Where in society have we achieved it? Can you get precision medical care? We have a public health approach to things. We made a huge advance last fall. We actually have female-sized crash test dummies now. Finally, that's how precise we are. So I think, we need to emphasize the accuracy, being directionally correct, probabilistically accurate at scale, doing no harm. That would be a good one. Trust that, yes, we have to use our best professional judgments, but we've also got to be out there policing ourselves, and we need a strategic framework. And I went too long, but I always do. I drove a long way.
Well, thank you very much for think about. So let's take a few minutes. Anyone would like to volunteer to ask a question? Sure. Yeah. We find common size.
Right.
The question for you is, has this changed the way you advise your graduate students how to experiment with testing, knowing that they
So we could use.
Okay. All right. So the question is, how has, the power analysis issue is a real issue, and has my sort of thinking about this changed how I'm advising my students and the types of experiments? Well, one challenge I have is I run this long-term experiment that was established in 1992, and it takes a lot of my time, and it has 4 replicates. I think what's changed is the way I talk to them about things like p-values and what you can expect in terms of effect sizes. We're having a discussion about this right now with one of our funders about does a particular nitrification inhibiting product, can we provide them with data that shows, because we can look at both, environmental and water quality impacts as well as crop growth. And the discussions I have with my students, and now even with the funder, is, look, you're looking for a very small effect size in amongst parameters that inherently are extremely variable. And there is a risk that the funder will say it's not worth funding that research. The conversation we were able to get to was to say, okay, you've done this elsewhere. You have other studies where you've done this. Can we combine the studies? We can show trends or whatever, and maybe we can't, but the likelihood that I am going to have in 1 or 2 years of research a significant result, given the variability in loads to, losses to drainage water and greenhouse gas emissions, highly unlikely given what we know about variability in these measurements. But I think it needs to be, oftentimes it's an affordability issue, right? You can't do 16 replicates. Or team with your colleagues. Are you guys doing this experiment? What do your treatments look like? Can we share data? Can we combine data? Can we do it, proactively?
Nothing yet.
Okay, question here.
The variability, especially with fish area and this is basically the end variability. Is variability we know about. Some of those variables we're just kind of purposely trying to ignore or use them to random effects. Any thoughts about?
Sure, and yes, the question was about covariates that might explain additional variability. And I should have mentioned that in the discussion of what I talk to my students about. We tend to try to collect as many of the covariates we know about as potential covariates as possible, so that you can try to parse out more of the error than you can if you don't collect some of those covariates. That said, you still, in some of these targets that we're interested in, and I think it was you, Mike, we're talking about, well, a better target for mitigation would be just reducing N applications, because in the grand scheme, you reduce N applications by 10%, some number, you're going to reduce environmental losses by some percent overall. And that some of that is, NTO emissions, some of it is nitrate leaching losses that may end up as indirect climate forcers. These are good things to try to understand, but and understand the science behind them and understand the covariates. But in the end, when you're predicting or you're trying to predict what will happen at scale, you're going to have, probably fewer, covariates that you're trying to take into consideration when you're trying to say what works where and what effect can you see. I don't know if I've answered your question, but.
Okay, thank you. I think we're moving past our normal time. This has been a really very interesting hour for me. I'm sure that's been for you. Thank you all for attending today. Dr. Brouder is going to be here tomorrow morning.